And thank you everybody for coming and listening. So you might be wondering, why in the world is a ruminant nutritionist trying to talk about topics that most vets are probably interested in? Well, we have been seeing a lot of the traditional cover crops that people are wanting to turn cattle out on, that we can have some potential issues causing some toxicosis, other metabolic disorders in cattle. So I just wanted to bring up some of these areas or some of these plants and some management strategies to help be cognizant of. So first off, here is a list of all of the potential issues that these cover crops or annual forages can have for cattle or other species. I might talk about some sheep and horses and some of these other plant species later on, just in case you have producers looking at utilizing these forages for those species. Bloat, glucosinolates, grass tetany, nitrates, PEM, prussic acid, sweet clover, and some of these are just plain poisonous. We don't necessarily know what is the active ingredient or the active chemical causing issues for livestock, but we do know that it causes negative effects. Now, for some of these that you might not be as familiar with, like what is glucosinolate, I'll describe it in more detail when I get to the plants that have issues with glucosinolates. What is grass tetany, etc. So I first just want to talk about plants that we know have been identified as being a poisonous or toxic plant. These are actually listed on USDA poisonous plant websites, or listed on the USDA poisonous plant website as a livestock poisonous plant. The first one is hairy vetch. Okay, I know you're already sitting there going, wait a minute, I've had producers, I as a producer have been grazing hairy vetch and have never had any issues. Okay, not all animals have issues with hairy vetch, but the animals that do be affected by hairy vetch, mortality or death occurs in 50 to 100% of those affected animals. And typically, your black-hided or black pigmented animals are the most commonly affected. Your Angus cattle, black cows, or black horses, Holsteins, black-headed sheep, etc. However, there has been some reports of Hereford Ranch having a significant amount of hairy vetch toxicity in their cow herd. Now, interestingly, we've known that hairy vetch is a poisonous plant or a toxic plant for many years. However, we still don't really know what is the compound that causes some issues. There's been several guesstimations, for lack of a better word, that it might be calcium cyanamide that is the chemical in hairy vetch that causes issues. However, nobody's been able to identify how much is too much, when it causes issues, and whatnot. Some of the clinical symptoms you see is this dermatitis, or when cattle kind of get this hypersensitivity to sunlight, and then a lot of times by the time you see that issue, within overnight, a lot of them will die. If those who don't, like I said, mortality is between 50 to 100% for your affected animals. Death usually occurs due to some kidney failure, and other research, we've said possibly it's this calcium cyanamide that might be causing the toxicities in hairy vetch. Other researchers think that it might just be kind of an allergic type of a reaction, that certain cattle just are genetically predisposed to be allergic to hairy vetch. One of the best synonym I come up with is peanut allergies in humans. We might have hairy vetch allergies. And so some animals, they have a very, very slight allergic reaction, and others have a very severe one. Hairy vetch kind of concerns me from a grazing perspective, just due to the fact that we don't know what to test for in the plant to determine when it can be toxic to our livestock species. We can't test the animals to determine which ones are susceptible to it. So it's just kind of a roll of the dice. You can turn out some cattle and be perfectly fine one year. 4 years later, you might kill 2-3 head overnight. That's exactly what happened at our research center. We grazed some hairy vetch in fescue for several years, no problem. And then the next year, we had 3 cattle come down, or at about year 4, had 3 cattle come down with hairy vetch toxicosis. 2 of them passed away, and the other one, she survived but lost her calf. So hairy vetch typically affects your older cattle more than younger ones, but we still just don't know what causes the issue. Now, I don't know what is the susceptibility rate for hairy vetch toxicosis in cattle. I imagine it's fairly low. We don't see a lot of cattle infected by it, but when you do, it can be devastating. Another poisonous plant are your lupins. And these, I don't know that people are necessarily planting them directly for cattle grazing purposes, but they are suggested as a cover crop species. Now, there are multiple different subspecies of lupins that are non-toxic, but there are 6 that are very toxic. Your silky lupine, tall cup velvet, silvery, summer, and sulfur lupine. And these are images of the plants when they are in their flowering stage, from left to right and then down to your lower left corner and over. Silky, tail cup velvet, silver, summer, and then your sulfur is the yellow ones. So what symptoms do you see with lupin toxicity? Well, it's when you get cleft palates, crooked legs, malformed spines, and death in some animals. Most of the time, the lupin issues occur in calves born out of cattle that had grazed it while being pregnant. It's an alkaloid that causes the issue. And in the western United States, lupins actually grow wild in nature and actually at a fairly high degree. So they have quite a bit of lupin toxicity out in the western U.S. The seed stage is when the plant is the most toxic. Deformities typically occur when a pregnant cow is placed on the seeded lupin when they're about 40 to 70 days of gestation. And sheep are actually more susceptible to issues with lupins than the cattle are. Amaranth is another poisonous plant, or can be very poisonous to livestock species. It's a cousin to pigweed. And we know that pigweeds are nitrate accumulators, and we can kill cattle due to nitrate toxicity with pigweeds. Same thing with amaranth. Also, there are kidney failure issues that associate with it. Kind of with the same thing with the lupins, there are some species of amaranth that are probably okay, and other species that are very, very more susceptible to being toxic. Now, another issue with your amaranth, it's called a true weed. It's very hard to control in pastures and crop ground because it's glyphosate resistant. So you might want to, try and discourage folks from planting this, especially from a cattle grazing perspective, but also just from a general cropping systems perspective. As I said, some amaranth species that are used for grain production, the grain is quite possibly okay to feed. But like this green amaranth, in this phase is not very safe to feed. Very high nitrate can lead to death in your livestock species. So those are the ones that are concerning to me because we don't necessarily have great management practices in place to try and minimize our loss in performance or death loss due to those toxicities. These other plants that I'm about to talk about, we are concerned about some metabolic issues with them, but we do have some management techniques that we can mitigate and be able to help improve or help maintain production and still be able to use these high quality, often high yielding products. The plants that I'm going to talk specifically about are your brassicas, small grains, lagoons, and your summer grasses. Okay, so your brassicas, they have a whole list of potential issues for cattle in particular. The first one, they are very high moisture and low fiber, which typically just leads to loose stool, but it also increases passage rate, so you might not be maximizing cattle performance. Brassicas, they can range anywhere from 15 to 25% dry matter and about 20% NDF. Very, very low moisture or very low dry matter, low fiber. So, simple, easy way to help manage to improve production, just supply a dry feedstuff. Allow cattle to have graze stalks, corn or sorghum stalks, or provide a hay, or plant something that'll be going dormant at the same time as the brassicas will be growing. The other issue with your glucosinolates in brassicas are your glucosinolates. They're what are primarily causing that hot flavor in your radishes, you know, for what we have as horseradish. So they're really good to fight off insect, but at the same time can cause some issues in livestock. One of the biggest ones is it decreases mineral absorption, especially iodine, and which causes some thyroid issues. It's also very low in copper, manganese, and zinc. And when you interfere with other mineral absorption, especially those three, you can have feet and eye issues. Copper, zinc, and manganese are very important from an immunologic standpoint as well as a reproductive standpoint. So we have some issues with glucosinolate interfering with your mineral metabolism. One way to offset that, provide chelated or highly absorbable and palatable mineral. Oftentimes cattle really don't want to eat mineral on these brassicas as well. So if you're finding that your cattle are not consuming a free choice mineral, you need to offer your mineral in a daily feed supplement or a every other day feed supplement. Just some way to try and get them these highly absorbable, highly palatable mineral. Polioencephalomalacia, anemia, and emphysema are all potential issues with your brassicas if you provide them at 100% of the cattle's diet. If you keep your brassicas at less than 75% of the total diet, mix it with a grass or a hay or something like else, you minimize your issues with PEM, anemia, and emphysema. And then the final potential issue with your brassicas are nitrates. Brassicas are nitrate accumulators, and when you test them, they will come back very high in nitrate value, especially pre-freeze. Now you might see that I have several question marks following nitrate issues with brassicas. Well, according to our nitrate values and what we've been seeing being turned in here in Kansas, we should have cattle dead cattle all across the state of Kansas grazing these brassicas. But interestingly enough, we don't have many dying due to nitrate toxicity on your brassicas. I'm going to talk you through possibly why we're not seeing some of those issues and talk about, even if we do have high nitrate values, some management practices that we might be implementing that are not causing us to have nitrate issues. So from just the brassica perspective, if you allow the plant to mature, it'll have lower nitrate later in the season, and you slowly introduce your animals to your high nitrate plant. Allow the rumen microbes to be able to adapt to nitrate digestion, and you should minimize your issues for nitrate toxicity. So this data actually came from Dr. Doug Shoup, where in 2012, in that winter, which was a winter after a severe drought, he measured brassica nitrate levels as represented by the blue diamonds, and cereal nitrate levels as represented by cereal grain nitrate levels as represented by the green squares. In early or mid-October, you can see very, very high nitrate levels, much higher than the threshold for potential death. According to our recommendations, about 9,000 parts per million on a dry matter basis of nitrate is where you potentially can have death in cattle species. But by the first mid-part, end part of November, nitrate values of the brassicas and your cereals were below that 9,000 parts per million nitrate. Okay, another thing to look at here that a lot of people don't necessarily realize, while your cereal grains, cereal grasses, rye, barley, oats, triticale, wheat are growing, they have very high nitrate values. And from these numbers, we should have been having cattle dying due to nitrate toxicity all the way up to the first part of November. I have yet to see reports of nitrate cattle on grazing wheat pasture dying due to nitrate toxicity. So I think there might be some complexity or some variation within our nitrate recommendations for grazing of these green growing forages. So I'm going to spend just a little bit of time talking about just the nitrate toxicity perspective. Some issues with nitrates: they can cause abortions, decreases performance, and can potentially cause death. This table just shows the general interpretation for forage nitrate if you submit it to your testing lab, and this is parts per million of nitrate on dry matter basis. Depending on what lab you send your samples to be analyzed for nitrate, some report back as nitrate, nitrogen, some report as nitrate, so you just need to be cognizant of what that is and look at their interpretations to be able to identify where you potentially have some issues. Another thing that I want to go ahead and make sure I emphasize, these guidelines are very accurate for nitrate toxicity, abortions, and death on your dry forages, your hays, sorghum stalks, corn stalks. So if you're testing these dry forages, follow these guidelines. They are very accurate for it. So just to give you a brief physiology of how and why nitrate toxicity causes issues. Cattle consume nitrate, and in the rumen, there are rumen microbes that work very rapidly to convert nitrate into nitrite. And then in a perfect ruminal environment, there's another set of microbes that'll convert nitrite to ammonia, which is a fairly safe system of being able to convert nitrate into a usable source for the ruminant species. However, in nitrate toxicity issues, or when you have really high levels of nitrate, these rumen microbes that are very quick and very easily able to convert nitrate to nitrite start producing so much nitrite that these really slow rumen microbe bugs can't keep up and convert nitrite to ammonia. So then this nitrite, some of it can get flushed out of the rumen, but most of it gets absorbed into the bloodstream, where the nitrite ions bind with red blood cells to form a compound called methemoglobin. When they bind, the nitrite binds to the red blood cells, it decreases the oxygen carrying capacity of that animal, leading to some issues with rapid heartbeat, decreased body temperature, muscular trimness, ataxia, weakness, etc. They just can't carry as much oxygen as they need. As well, those nitrite ions can bind to the smooth muscle of the blood vessels and cause vasodilation, which further restricts blood flow to the animal. So that's why at lower levels of nitrate intake, we can cause abortions. If you decrease the amount of oxygen, decrease the amount of blood to the fetus, you're more likely to cause a fetal abortion. Now, the nitrite, it's actually 10 times more toxic to the animal than the nitrate itself. But we test for nitrate. It's a more stable chemical to measure in our plants. So that's where it's not nitrate that actually kills cattle. It's the nitrate. It's nitrite itself. So what factors are involved in determining whether we have nitrate toxicity or not? And why have we not been seeing cattle that were on 25,000 parts per million brassicas not even having abortions? Well, I found a study from 1979, the Netherlands Journal of Agricultural Science, and they had 40 different experiments using the dairy cow as the model, where they fed hay, grass pellets, turnips, or fresh mown grass to these dairy cows, and looked at formation of methemoglobin. So I first want to show you this one figure from their article. They have nitrate intake along the x-axis, and then they have methemoglobin as a percentage of total hemoglobin along the y-axis. Now, just to kind of give you a point of reference, when you're about 30% methemoglobin, that's when you start seeing clinical signs. That rapid heartbeat, muscular tremors, weakness, etc. At about 50%, that's when their membranes will actually go from being pink and healthy looking to a brown or blue cyanosis type of appearance, and death occurs at 80%. So they actually didn't cause death in any of these animals in their study, but they were getting clinical symptoms as occurred at about 30%. Now there is there's open circles, X's, and dark circles, or filled in circles in this figure. very hard to see, so I split this up, and for the hay, at lower levels of nitrate, you started getting detectable levels of methemoglobin in the blood, especially up to showing clinical signs of nitrate toxicity. Your turnips were more concentrated at a higher level of nitrate intake is when you started seeing increases in methemoglobin. And then your fresh mown grass, which are most of these dark circles, you had to have much higher levels of nitrate intake before you started seeing detectable levels of methemoglobin and all the way up to clinical symptoms. Another interesting part of their study is they looked just at hays, and they found that around this, and I'm sorry I didn't convert this to units that we're more familiar with, but at about a 12 gram per 100 kilograms of body weight is when they started seeing clinical symptoms. But look at the range of methemoglobin at the 12. You have everything in hay from less than a 5% all the way up to about a 60% methemoglobin in the blood. So just because there's a specific nitrate amount or a specific nitrate intake, total nitrate intake, that didn't completely answer the question of what should our threshold be. So these researchers decided, okay, we're going to feed them the exact same amount of nitrate, but we're going to either make them eat it in 25 minutes or, and they fed them 30 pounds of hay. They either made them eat all 30 pounds in 25 minutes or spread it out into 8 feedings through the day for a total of 110 minutes of intake. With the hay, at that 20, as represented by the X's, and I am trying to get, okay, here we go. At these X's, you can see there's much higher methemoglobin levels in the hay that was consumed rapidly versus the hay that was consumed slowly. So then they thought, okay, well, we'll look at the grass pellets. Do we see that same trend? So once again, when they ate slowly, they had much lower levels of methemoglobin, whereas if they ate faster, they had higher levels of methemoglobin. However, if you look at the, our 30% cutoff as showing clinical symptoms, your hay, you were much closer to showing your clinical symptoms versus the grass pellet. And this was at a lower nitrate level. At a higher nitrate level, once again, your hay started showing you some clinical symptoms, whereas your fast hay intake, your fast grass intake, I am so sorry, you started showing clinical symptoms. Slow intake, not so much. So, bottom line, the faster they consume high nitrate, the greater chance you have of showing nitrate toxicity symptoms, regardless of whether it was a hay or a grass. Now, another thing that I thought was interesting from their study, and this one's a lot easier to understand, they just put hay is represented by the open circles, turnips is represented by the X, and fresh mown grass is represented by the black circle, incubated them just in distilled water, and measured how much nitrate was diffused into the water across time. Within about 30 minutes, nearly 80% of the hay was diffused into the water, whereas only 30% for your turnips and only about 10% for your fresh mown grass. After 3 hours, your hay had about 95-ish% of the nitrate diffused into the water, only about 30% for your turnips, and only about 10% for your fresh mown grass. So this demonstrates that there might just be a plant physiology component that the higher moisture plants might not diffuse as much of the nitrate into the rumen environment. Therefore, your rumen microbes might be more readily able to keep up with the nitrite to ammonia conversion. So, nitrate toxicity, as you could tell, is a very complex issue. Can be impacted by the amount of nitrate consumed per meal, the speed of intake, and quite possibly the rate at which the nitrate's released from the plant. So when you think about all of these added together, even though the brassicas are very, very high in nitrate, they're also very low in dry matter. So they have to eat a lot of brassicas to be able to eat an equivalent amount of nitrate from a hay, essentially 3 times as much. And therefore, if they have to eat 3 times as much, on wetter feeds, they eat just a little slower, so your speed of nitrate intake is slower. And if the brassicas just don't release much nitrate, that might be why we weren't seeing issues with grazing brassicas, or why we don't see nitrate toxicity issues on your wheat pasture. Some other things that we can do just to manage for high nitrate feeds. Dilution is the solution to nitrate pollution. So you can dilute by having good, clean water. Dilute by providing another source of dry feeds. Provide energy. There's been several studies that show that if you provide some extra corn, those rumen microbes that convert nitrite to ammonia, they can keep up to a higher degree with some extra energy. And sulfur actually can help those rumen microbes that convert nitrite to ammonia. So providing a mineral that has and the appropriate amount of sulfur can help manage for your nitrate issues as well. So that was a lot of information about nitrate toxicity, but that was something I felt like we needed to talk about because our current testing for nitrate is very accurate, and it is the guidelines for use of these high nitrate feeds for dry forages is very accurate, but we might have a little more wiggle room for our high moisture feeds. I don't know exactly what our guidelines should be as of yet. We have a couple of research projects that we're trying to answer that question. So if you're completely risk adverse and you test and you have high nitrate green forages, don't feed it. If you're willing to take a gamble, we might be able to feed a little bit higher levels, as long as it is green and growing. Okay, so that was the component about the did have a comment from Mary saying if sulfur helps with managing for nitrates, and she says, and there's high sulfur in brassicas, that might be part of the reason why cattle handle nitrate toxicity better, because brassicas are usually between 0.5 to 0.7% sulfur. That could be one of the areas I didn't even think about, but that would make sense because sulfur has been shown to help with nitrate toxicity. And also, to give you another list, I should have put this on the slide, but other high nitrates feeds, according to the Merck Veterinary Manual, your cereal grasses, especially oat, millet, and rye. Corn can be high in nitrate, sunflower, and sorghums. Your weeds that are very high in nitrate and can cause issues are pigweed, lamb's quarter, thistle, jimsonweed, fireweed or kochia, smartweed, dock, and Johnson grass. Okay, I have another question. It says, ask, do I know of any situations where Palmer Amaranth caused nitrate problems? What do you think about grazing with Palmer present? If you think grazing growing plants are not a problem. You potentially, and with my amaranths, I'm sorry, I'm jumping back through here. Lupins. I'm not exactly sure. I'd have to look up because I haven't worked directly with Palmer to see whether it is one of them who could potentially cause issues with other Palmer amaranth as by itself. In general, the amaranths are high nitrate, and you know, okay, I can see where your question's going, because Johnson grass, when it's green and growing, is high nitrate, and we know we kill cattle on green growing Johnson grass, and we know we can kill cattle due to nitrate toxicity on green growing pigweed. So your Palmer amaranth, those ones might be some exceptions to the rule, because we do know that those, you can kill cattle with nitrate on those green and growing. Your brassicas and your cereal grasses, we don't seem to be having that same issue. And by the time we're done with this, you're probably you're gonna have more questions about what we should be doing for nitrate on the green growing forages than what you had before, because I think that is an area that we need more information on. Okay. Switching over to my grasses, we have your barley, oats, rye, ryegrass, wheat, triticale. In the fall, winter, and spring, when they're really growing and lush, you could potentially have issues with grass tetany. Pretty easy to manage. Just have a high mag mineral, somewhere in 8 to 12% mag, or you just turn out your non-lactating cattle. A grass tetany primarily impacts cows that are lactating, especially in early lactation. So if you want to graze something that potentially can be low in magnesium, which is, grass tetany is hypomagnesia, low magnesium, just turn out cattle that aren't as sensitive to magnesium, such as your stalkers. And that's why Oklahoma has made a great living on turning out stalker calves on wheat pasture. Another thing, if you did want to put your lactating cattle out on these grasses, you can include in a lagoon at about 30% of the pasture. Those lagoons will kind of help manage for your grass tetany. Now, if you remember my talk from a month or so ago, in Kansas, we have a little bit of an issue trying to get our lagoons established and growing in the fall. So you might include a lagoon in the form of a hay. But the easiest management practice is just provide your high mag mineral and put a non-lactating animal on these. Bloat is also an issue. But once again, we have a lot of research and a lot of management practices that we can use to help minimize bloat issues on these grass species. Provide a dry forage. Provide ionophores and bloat blocks. Then once again, nitrates. And even the vet manual says that oats and rye are especially high in nitrate. So that's a potential issue. So put out your sulfur. In regards to your lagoons, the number one issue is bloat. Now, we do have some non-bloating lagoons, Lespedeza, bird's foot trefoil, and sainfoin. One thing I did want to bring up, I've seen a couple of reports now that says bird's foot trefoil has issues with prussic acid. So if you're completely risk adverse, you might not want to include bird's foot trefoil. And another issue in your lagoons, your clovers. One of the most common issues we recognize is sweet clover poisoning, but that happens with moldy hay is the most common culprit for sweet clover poisoning. And the active ingredient or the active chemical that causes issues with sweet clover poisoning is coumarin. And that can be found in sweet clover, yellow clover, and white clovers. Your red clover has the lowest coumarin content. So if you're potentially wanting to put up something that has clovers or put up your clovers as a hay, your red clover is your least susceptible one. But if you put it up nice and dry and don't let it get moldy, you pretty well minimize your issues. And then your sorghums, sedans, millets, and corn. Prussic acid is something that can be an issue. Your sedan grass has the lowest issue. Sorghum sedans, intermediate, and grain sorghums are the highest. We do not have prussic acid issues in corn, pearl millet, or foxtail millet. Now, a lot of times we talk about prussic acid and nitrate issues as one and the same, mainly because sorghum sedans, millets, and corn all might, that broad category can potentially have both. Well, prussic, just because your plant might have prussic acid doesn't necessarily mean it has high nitrate. Just because it has high nitrate doesn't necessarily mean it has prussic acid. There are definitely differences in what causes issues with your prussic acid. It's a cyanide toxicity, and your prussic acid, once the plant is dead, be it harvested as hay or has gone into dormancy, the prussic acid will actually dissipate via some evaporative processes, and prussic acid goes away and is no longer a concern. Nitrate, on the other hand, unless you're ensiling the plant, once it is dead, your nitrate values stay fairly static. So that's just kind of, a little bit about what's the difference in prussic acid versus nitrate. Now, we do have some rules of thumb to help manage for prussic acid issues. if you graze your millets, especially when they are taller than 18 inches, you should minimize your issue for prussic acid. And also, because they regrow in about 5 days, if you use a rotational grazing system, only graze your millets for about 5 days so you don't have your cattle consume the regrowth. Prussic acid is much higher in your regrowth and drought-stressed, or in your regrowth and immediately damaged due to immediately damaged plants. I already mentioned that your sorghum, sedans, millets, and corn can all have high nitrates as well. Ways to manage for that: allow the plant to mature. This information from Dr. Holloman out in western Kansas shows that as the plant matures, it'll decrease, but also if you increase your cutting height, you will start decreasing the amount of nitrate in the hay that you are cutting. Most of the nitrate will be accumulated in the lower stem portion of your sorghum, sedan, millet, or corn. That same rule can be applied when you're grazing your sorghum, sedans, millets, and corn. To minimize for prussic acid issues on sorghums, you wait until they're about 2 feet tall, and then you graze them. To minimize some nitrate toxicity issues, you graze it until there's 8 inches left. In theory, most of your nitrate should be left in that lower 8 inches of your plant, so you shouldn't have nitrate toxicity issues. As well, if you leave 8 inches on your sorghum, that is what they recommend to allow the plant to regrow. And also, for the sorghums, you can be in that paddock for about 10 days before you start getting too much regrowth, green, to potentially have prussic acid issues, etc. And in general, if you have about 25-day rest period, can get you back up 24 inches, assuming you have appropriate moisture, fertility, etc. And this is a very rough estimated stocking rate, but to best be able to determine your stocking rate, you need to measure how much forage is out there. For your sedan and millets, because they're just slightly different than your sorghums, you can graze them when they're a little shorter. Once again, to minimize nitrate issues, leave 8 inches because they grow much faster. You have only graze for about 7 to 10 days, and rest period is about 21 days before they get back up to the 18 inches. They aren't going to yield, in general, as much dry matter as your sorghums do, so you have to stock a little lighter. All of this information, if you want more detail about specific forages, including a handy little table that shows you some estimated protein and energy and whether they're toxic and what species it might be toxic to, you can go to the K-State bookstore or just Google MF 3244 K-State, and all this information is in a downloadable publication. And for if you wanted to spend some more time calculating how many paddocks and estimating dry matter intake and what your stocking rate for a rotational grazing system for your sorghums and millets, you can find all of those equations can be found on the Animal Science website in the May 2015 Beef Tips. And I'm going to go ahead and give you guys another blurb. If you haven't went and played around with this tool, it's the Midwest Cover Crops Council. For Kansas, you can input what county you live in and what your, potential cash crop, etc., your soil types, but more importantly, you can input what your goal is, and if your first goal is to have a high quality forage, you can select that and it'll give you a list of forages that those of us in Kansas believe would be a valuable cover crop and forage for cattle. So it's a very handy tool. If you didn't get this address wrote down, you can just Google Midwest Cover Crops Council decision tool, and you will get right to this link. Thank you for your time, and there's my contact information, minus the phone number. For some reason, I put down the wrong phone number, but my correct number is 620-820-6125. And I'll open the floor for any questions you might have, which I do see one here. Neil asked, how can we tell in the field when sorghum Sudan hybrids are under stress, especially drought stress, and could be a prussic acid problem? An easy answer was by not saying it depends, which is the typical extension answer. If your sorghum Sudans are not taller than 18 inches, you probably had some kind of stress, and prussic acid might be a problem. You can test for prussic acid, but I told you that when you cut the plant and it starts dying, the prussic acid starts evaporating rather quickly. You can test for prussic acid, but you have to clip your sample, put it in an airtight bag, and get it to the lab immediately. Don't let it get hot. Don't let it get a lot of extra damage to it, etc. But one of the easier field tests is just if it's short, you might have a prussic acid issue.
All right. Well, if there aren't any other questions, I'll give it maybe one more minute. I just want to thank Jaymelynn so much for presenting for us this month. The full video of the webinar will be available on YouTube and on the project webpage either today or tomorrow, depending upon when we get that done. And I will send out an email letting everyone know when that is the case. If there's no other questions, thank you all so much for joining.